signals in voxels all over the brain. Now, you might get total nonsense. I mean, this might be just an exercise in fishing. I mean, the map might look like random numbers. And also, if you take the top or the best SNP, you're immensely inflating your false positive rate. So rather than using a standard uniform distribution for your p-values, you have to use something called a beta distribution. The beta distribution is the null distribution for the minimum of n different uniform distributions. And so if you're going to allow yourself to take the best p-value after 500,000 tries, you've got to guard against having null results pretty severely, and you use this beta distribution to test. And one of the things that this does, it leads to a lot of data. So that point there, I don't know if you can see that, that point in the image, if you're looking at gray matter density or functional activation there, each point has a little Manhattan plot in it. I mean, you can imagine a fantastic amount of data, and you take out the top p-value from your Manhattan plot, plug it back into the image, and see if there's any evidence of gene effects at all. And unfortunately, the answer is no. So one of the problems with doing this is, I mean, you just have an astronomical search space where you have 500,000 SNPs, a fifth of a million voxels, 138 billion tests, 10 days to run. And some of these genes, I mean, you can list them and find out what they do. They have plausible biological functions. But it doesn't beat the p-value threshold that you need to guard against the 138 billion tests. You'd have to have something like 10 to the minus 12. But you can do a little better than that. So nobody would do mass univariate testing in functional MRI. So we know that gene action in the brain is coherent. And pretty much any signal in the brain is somewhat coherent. If you know the value at one voxel, you've got a pretty good guess at what's happening at the next voxel. So what if you said, well, I think that any genes in the brain probably affect certain structures or certain systems, or they might be a diffuse pattern of effects. And instead of just testing each of these points as if they were in glorious isolation, let's use some of the spatial covariance or some of the spatial correlation structure in the image to give us a better chance of finding the genes. And one of the ways of doing this is a paper in Journal of Neuroscience, I think a month ago. He was my student, he's now a professor, Ming-Chang Chiang in Taiwan. He asked the question, would it boost the power of genome-wide scanning and imaging if you could cluster out of the images regions of the brain that had similar genetic determination? Now, I've got to say what I mean by that. How would you find a chunk of image that has similar genetic determination? Now, the answer is yes. It's really very useful. So I'll tell you how it works. It's all in his paper, but it's a little complicated. I'll take it step by step. So there is something known as genetic correlation. We're all familiar with correlation. So, shoe size and height are correlated. That would be known as phenotypic correlation. But let's say you had a twin. Maybe some of you do have twins. Let's say your IQ was linked with the brain size of your twin. Now, that's kind of a strange question. You'd ask the question, is the IQ of one twin linked with the brain size of the other twin? And that's called a cross-trait design. Well, usually it is, and so there is a correlation there. If that cross-twin correlation is higher in identical twins than in fraternal twins, you can blame it on the DNA. So this is something that's called a cross-twin, cross-trait design to estimate genetic correlation. Now, it's a very strange idea that you could use a trait in one twin to predict the trait in the other twin. But if you're better able to do so when the twins are identical than when they're fraternal, and identical twins have more DNA in common, then you can say that there's a genetic connection that's causing that phenotypic correlation. And that's called a genetic correlation. So you can measure the genetic correlation between all sorts of things, size of the caudate and size of the brain, or IQ and size of the brain. I mean, and these things are correlated, but you want to know, is there any genetic basis for the correlation? Is it just a correlation that comes about because things scale in a certain way as parts of the body? Or are there common genetic regions of the genome that are responsible for these traits or measures being correlated? Now let's take another layer of generalization. Let's look at an image. So let's say you're at one voxel in one twin's image, and you try and predict another voxel somewhere else in the brain from the other twin's image. Now this seems like a really strange thing to do. And you're using the fact that there's a genetic connection in their DNA to make the basis for your inference between one location in one twin's brain and a different location in another twin's brain. And like I said before, if you're better able to do that type of different voxel correlation or prediction in identical twins than fraternal twins, and you have to scan both kinds, then you have evidence of a genetic correlation. So in much the same way as you do resting state functional MRI, and this is probably a more familiar analogy, you can sit in a resting state scan and pull out all the voxels that have a synchronous time course. And that is a map of the resting state default mode, for example. You sit at a certain location and say, tell me all the other parts of the brain that have a functional synchronization with this voxel. In the context of genetic correlation, I mean, genetic correlation is just a different kind of correlation from resting state functional MRI. You could sit at a voxel in the brain and say, tell me all the other voxels in the brain that have a genetic correlation with this one. Now, the way we measured it was really strange. We used twins, we used different parts of the brain. But you could seed a voxel and pull out a whole hunk of voxels that have a common genetic correlation. And when the genetic correlation is high, the same bits of your DNA are contributing to the variations that you see in those different areas. So this was the basis for Ming Chang's method. It's called genetic clustering. And if you cluster voxels based on a genetic correlation, there's a direct analogy with resting state functional MRI, where you're clustering voxels into modes or systems. And the question is, if you run GWAS on these new ROIs, these clustered genetic ROIs, are you better off? And you are just fantastically better off. And so this is obviously the approach to take. And actually, many genome-wide significant hits were found in a data set of size 472 people. And not only that, whole networks of genes were pulled out. What do I mean by a network of genes? Well, rather than just saying, well, hey, we discovered a gene that affects hippocampal volume, you can build an entire network of SNPs that are correlated in their effects on different structures. So let's say you have a pair of genes, and each of them have correlated effects. They affect similar things in the brain. You can start to build a network where the spokes in the network are the degree of affinity between the gene effects on different structures. So as you can see, there's a lot of analogies going on here. So to get genetic correlations, which you need to pull out coherent gene effects from images, we're using an analogy based on functional resting state imaging. And to build these gene networks, we're using an analogy very, very similar to connectomics, where you ask the question, do these SNPs affect similar things? And if they affect a lot of the similar things, you give them a spoke in the network. So this is really becoming a very interesting area where connectomics is coming into genetics as well. So let's move on. Here we go. This notion of genetic correlation seems to be a savior to the field of imaging genetics. So when I asked the question, do you need 20,000 people to discover genes that affect brain traits, it was beginning to look like in April of this year that you did. And the reason is, only when we got to 21,000 people was there a genome-wide significant hit in that huge ENIGMA study. But only 2 months later, there's this paper in Journal of Neuroscience. And I have to say there's a group at UCSD led by Anders Dale, also had a similar idea of genetic clustering. And what you're beginning to see is a movement in the handling of brain images that's becoming much more multivariate. And so rather than the sort of early days of SPM, tell me if any voxels are linked with a paradigm, and the early days of genetics where you say, well, tell me if any jigsaw pieces of the brain or any voxels are linked with genetic variation, you're seeing a much more multivariate view where you're trying to pull out a pattern from the image that is coherently associated with one gene or a network of genes. Now this gives you a little bit of a leg up. And so if you're a mathematician, you often try and prove new things based on what you already know. And so you often take a theorem, I don't know, maybe Parseval's theorem in function analysis, and you'll use that to make assertions about Hilbert spaces or signals and things like that. So now that we've noticed that genetic correlation is very helpful to get at effects that were almost too small to detect, you can look at genetic correlations between blood markers and images. I'll tell you why that's reasonable to do. There's a whole load of genes that link with things you can measure in the blood, like cholesterol, folate levels, iron levels. I'll give you an example. That iron levels in your blood today will depend on a gene, the HFE gene, many of us have polymorphic variants of this, that tell you how much of the iron is transported into the brain and around the body. And this is the commonest genetic disease in the world, hemochromatosis. It results from mutation in the HFE gene. So you could begin to ask the question, is there a genetic correlation between measures in the blood, such as measures of iron and brain structure, for example, myelination on DTI? And if there is a correlation between those, you can start to look for gene effects in the brain. So there's been many studies like this. This is one by my student Neda Jahanshad, where she found that an iron gene basically was related to patterns of connectivity in DTI. But it was the mathematical principle that matters. It's not so much, I mean, iron story is interesting. What was known was that there's a correlation between iron levels in the blood and variants in this gene. Same happens with cholesterol and other measures. And then we proved that there was a link between iron levels in the blood and FA on DTI. And iron's a constituent of myelin, which makes a lot of sense. And then you can sort of join the dots together to try and find pathways that basically give you links between common genetic variants and variants in images. All right, so I'm going to segue in the last couple of minutes into Van's talk. I think Van's already here. So I thought I'd show a beautiful image, because he'll be showing lots of beautiful images of DSI. So you could say, well, if you're a gambler, would you bet on MRI, structural MRI, as a good treasure chest of genes that affect the brain? Or would you look at the connectome? And if you would look at the connectome, do you want to look at DSI, like Van is going to talk about, or would it be functional synchronization? I mean, what are the best brain traits to look at? And this is an extremely interesting question. So this is tractography, as you know. I know you've heard about this a little bit from Jeff and others on Friday. If you think of the detail that's encodable in a single scan, and these are Hardy scans, it's a variant, sort of simpler version of DSI where you have very high angular resolution of the scan. You can resolve all the fiber crossings, and I think Van's going to talk about this in a minute. And you can basically ask the question, tell me what fraction of fibers interconnects brain region A with brain region B. And not just what fraction, what is their fiber integrity, their FA, what is their mean diffusivity. And you guys know about this. You can basically parcellate the cortex into 70 or 1,000 regions, however many you want. And you can count these fiber connections, put them in a big matrix. And you guys have probably seen these already, where there's sort of N ROIs and N ROIs. And the stuff in the matrix is a new image with some quantitative index of what the wiring is between region A and region B. So you could begin to do genome-wide screening of the connectome. So that's a sort of exciting idea. So the connectome is already a very complicated thing. Although most connectomes, although they're displayed as graphs, can be represented mathematically as n by n matrices. So as you know, all these nodes that sit there being connected to each other, there's always a weighted link or nothing. And the weights on those links can be stored in a matrix. Well, an n by n matrix is just a 2D image. So you can throw all those 2D images, those connectomes, into this big grinding machine to look for genetic association. And this is a really exciting discovery. One of my students, Neda Jahanshad, has been looking at this. If you have a matrix of connections that's 70 by 70 or 4900, and if you screen the genome for hits, you've got a false positive rate of 0.05 times 500,000 SNPs times the 4900 connections. But one of the things that you see is it's very, very powerful. And you even, those sort of little hurdles. I guess it's the Olympic Games in a couple of weeks, so there's going to be a lot of bars that you have to get over. That bar that you see there, there's a whole bunch of genetic variants that exceed the bar that's necessary for them to be considered associated with that brain trait. And these are p-values of about 1 trillion to 1. So the reason I'm telling you this, and one other exciting thing is the SNPs that came from this study were later found in a different cohort to be Alzheimer's disease risk genes. So you can see that there's sort of a practical value to screening connectomes for genetic influence and then looking at what you get and going to other cohorts who are ill and seeing if there's any measurable impact on their clinical status. It's a very, very exciting direction of work. Now, if you had to bet on whether connectomes or structural images or functional images were good traits for genetic analysis, all of your